This slide shows how glucocorticoids such as cortisol exert their effects at the molecular level through intracellular receptors. Because glucocorticoids are steroid hormones, they are lipophilic and they can diffuse across the plasma membrane. Once inside the cytoplasm, they bind to the glucocorticoid receptors, which is initially held in an inactive state by heat shock proteins, or HSPs. Ligand binding causes a conformational change in the receptor. The heat shock proteins dissociate, and the activated glucocorticoid receptor complex translocates into the nucleus. Inside the nucleus, the receptor can regulate gene expression. This happens through two major mechanisms, shown here as transactivation and transrepression. In transactivation, glucocorticoid receptor dimers bind directly to specific DNA resequences called glucocorticoid response elements, or GREs. This leads to increased transcription of target genes, many of which are involved in metabolism and glucose regulation. In transrepression, the glucocorticoid receptor does not bind DNA directly. Instead, it acts with other transcription factors, labeled TF, such as NF-kappa B or AP1, and suppresses their ability to promote gene expression. This mechanism is primarily responsible for the anti-inflammatory and immunosuppressive effects of glucocorticoids. The key idea is that these two pathways are mechanistically distinct but occur simultaneously. This is why glucocorticoids can both control inflammation and disrupt metabolism at the same time. From a medicinal chemistry perspective, this distinction is critical. Many modern glucocorticoid drugs aim to preserve transrepression while minimizing transactivation, in an effort to retain therapeutic benefit while reducing side effects. This slide zooms in on the glucocorticoid receptor itself, showing how protein structure is organized into functional domains. Understanding this architecture helps explain how a single receptor can produce such diverse biological effects. Starting with the N-terminus, the receptor contains a large N-terminal domain, or NTD. This region is involved in transactivation and contains activation function 1, or AF1. Importantly, this domain is highly flexible and interacts with coactivator proteins that regulate gene expression. In the middle is the DNA binding domain, or DBD. This region is responsible for recognizing and binding specific DNA sequences known as glucocorticoid response elements, or GREs. It also plays a key role in receptor dimerization, which is required for certain gene regulatory events. Adjacent to the DBD is the hinge region, labeled H, which contributes to nuclear localization and provides structural flexibility as the receptor moves and rearranges inside the nucleus. At the C-terminal domain is the ligand binding domain. This is where cortisol and synthetic glucocorticoids bind. Ligand binding to this domain controls receptor activation, interaction with heat shock proteins like HSP90, and recruitment of transcriptional cofactors through activation function 2, or AF2. The key takeaway from this slide is that the glucocorticoid receptor is not a single functional protein. Different domains control ligand binding, DNA interaction, nuclear localization, and transcriptional outcomes. Small changes in ligand structure can shift how these domains interact with one another, leading to very different biological effects. This slide shows the major mechanisms by which the glucocorticoid receptor regulates gene expression once it has entered the nucleus. The key point is that glucocorticoids do not simply turn genes on or off. They regulate transcription through multiple mechanistically distinct pathways. In the top left, we see direct DNA binding, also called transactivation. Here, the glucocorticoid receptors form dimers and bind directly to the glucocorticoid response elements, the GREs. This typically leads to increased transcription of target genes, many of which are involved in metabolism and glucose regulation. In the bottom left, we see direct DNA binding to negative GREs or NGREs. In this case, receptor binding leads to the suppression of gene expression. This shows that even direct DNA binding does not always result in activation. Context matters. On the right side of the slide are two indirect mechanisms. In composite regulation, the glucocorticoid receptor binds DNA near other transcription factors. The combined presence of glucocorticoid receptors and those transcription factors can either increase or decrease gene expression, depending on the cofactors recruited. Finally, in tethering, the glucocorticoid receptor does not bind DNA at all. Instead, it interacts with other transcription factors, such as NF-kappa B or AP1, and alters their ability to regulate transcription. This mechanism is central to the anti-inflammatory effects of the glucocorticoids. The key takeaway is that the glucocorticoid receptor is a context-dependent regulator. The same ligand-receptor complex can activate, repress, or fine-tune gene expression, depending on how it engages DNA and other transcriptional machinery. This figure summarizes the broad immunological effects of glucocorticoids such as cortisol across both innate and adaptive immune cells. Starting at the top, glucocorticoids act on the vascular endothelium to reduce expression of adhesion molecules. This limits neutrophil rolling, adhesion, and migration to inflamed tissues, effectively reducing the early inflammatory response. In innate immune cells, like monocytes and macrophages, glucocorticoids suppress the production of inflammatory cytokines, chemokines, and reactive oxygen species. They also reduce phagocytic activity and promote apoptosis, further dampening inflammation. Dendritic cells are also affected. Glucocorticoids impair dendritic cell maturation and migration and reduce their ability to present antigen and provide co-stimulatory signals to T cells. As a result, downstream T cell activation and differentiation are altered. Glucocorticoids suppress pro-inflammatory T helper subsets, such as Th1, Th17 cells, while favoring more regulatory or anti-inflammatory phenotypes. The key takeaway is that glucocorticoids do not target a single immune pathway. Instead, they act at multiple levels of the immune system simultaneously, reshaping immune responses in a coordinated way. This systems-level immunosuppression explains both the therapeutic power of glucocorticoids and the risks associated with their chronic use. This diagram shows what mineralocorticoid receptor activation looks like in a kidney principal cell. Aldosterone, a related steroid hormone, diffuses into the cell and binds the mineralocorticoid receptor, and the receptor ligand complex translocates into the nucleus. This leads to transcription of genes that increase sodium reabsorption through ENaC on the apical membrane and enhance sodium transport through the sodium-potassium ATPase on the basolateral membrane. The net effect is sodium and water retention coupled with potassium excretion. Importantly, cortisol can also enter this cell, but it's rapidly converted to cortisone by the enzyme 11-beta hydroxysteroid dehydrogenase type 2. Cortisone does not activate the mineralocorticoid receptor, which protects the kidney from chronic cortisol signaling. This protective mechanism is central to understanding both normal physiology and the side effects of glucocorticoid drugs. This slide shows how cortisol signaling is regulated at the tissue level by 11-beta hydroxysteroid dehydrogenase enzyme. In the kidney and placenta, 11-beta HSD2 converts cortisol into cortisone by oxidizing the 11-beta hydroxyl group. Cortisone does not bind to the glucocorticoid or mineralocorticoid receptor, effectively inactivating cortisol in these tissues. In contrast, the liver expresses 11-beta HSD1, which catalyzes the reverse reaction, converting cortisone back into cortisol and amplifying glucocorticoid signaling. This enzyme system allows the same hormone to have very different effects depending on the tissue, and it plays a central role in determining both therapeutic effects and side effects of glucocorticoid drugs. This slide shows the chemical structure of cortisol, the primary endogenous glucocorticoid in humans. Cortisol is a 21-carbon steroid with the molecular formula C21H30O5. Like all steroid hormones, it's built on a 4-ring fused backbone derived from cholesterol. You should be able to recognize steroid backbone structures. This rigid scaffold is essential for high-affinity binding to steroid hormone receptors, including the glucocorticoid receptor. Several functional groups on this structure are especially important for biological activity. The hydroxyl substituents are critical for hydrogen bonding interaction within the receptor binding pocket. As we just saw with cortisol inactivation in the kidney, changing even a single hydroxyl group can switch the molecule from an active to an inactive state. The carbonyl groups also contribute to the receptor binding by influencing the molecule's electronic distribution, while the largely hydrophobic steroid core allows cortisol to readily diffuse across the cell membranes. Importantly, cortisol's three-dimensional shape, including the orientation of hydrogens above or below the plane, determines how the molecule fits into the receptor. This stereochemistry is not optional. Altering it can significantly reduce or completely abolish biological activity. This structure serves as the natural reference point. All synthetic glucocorticoid drugs are chemically modified versions of this scaffold, designed to enhance potency, alter receptor selectivity, extend duration of action, or reduce unwanted mineralocorticoid effects. This slide further depicts the structure-activity relationships, or SAR, of cortisol with the glucocorticoid receptor. We'll start with the A ring on the left side of the molecule. The 3-keto functional group and the delta 4-5 double bond are essential for both glucocorticoid and mineralocorticoid activity. Removing either feature dramatically reduces receptor binding and biological function. Together, these elements form part of the core recognition motif shared by steroid hormone receptors. Moving upward, the 11-beta hydroxyl group is required for glucocorticoid activity. This hydroxyl participates directly in receptor interactions, and its presence distinguishes active glucocorticoids from inactive precursors such as cortisone. Introducing a delta 1-2 bond between carbons 1 and 2 increases glucocorticoid activity. This modification is commonly used in synthetic analogs to enhance potency relative to cortisol. On the right side of the molecule, the carbon-21 hydroxyl contributes to mineralocorticoid activity, although it's not an absolute requirement for glucocorticoid effects. Modifying or masking this position is one strategy used to reduce unwanted mineralocorticoid side effects, such as sodium retention. Substitutions at carbon-16 tend to decrease mineralocorticoid activity. This is a key medicinal chemistry principle. By adding steric bulk or polarity at C16, compounds can be biased towards glucocorticoid selectivity. Finally, halogenation at positions 6 and 9, most commonly fluorination, enhances overall activity. Fluorine increases receptor affinity and metabolic stability, which is why many high-potency synthetic glucocorticoids include a fluorine atom. The takeaway from this SAR map is that cortisol serves as the baseline structure. Medicinal chemists tune potency, selectivity, and side effect profiles by making precise modifications at a small number of strategic positions. In the next slide, we'll see how these principles are applied in real drugs such as prednisone and dexamethasone, and why those structural differences matter clinically.